Hello, everyone, and welcome to webinar Link Prediction for Biomedical Innovation and Drug Discovery. So the goal today is to learn fundamentals of training your own knowledge graph embeddings and use them for link prediction tasks and apply it to drug discovery. After this webinar, I hope this computational method will feel less inaccessible to all of you, and you will feel more empowered to apply this method in your own research. I'll start with a mandatory disclaimer that QIAGEN products shown in this webinar are intended only for research and molecular biology applications. So agenda for today is as follows. I'll start with a brief introduction of QIAGEN Biomedical KBHD, our knowledge graph of human curated biomedical relationships. We will use this knowledge graphs as an example for demonstrating how to train embeddings. So this is the data underlying all application of link prediction will be based on biomedical KBHD data, and that will also be used for evaluation. Then I will continue with a short overview of theoretical landscape behind link prediction methods. And then we get practical, where I will try to convince you that how easy it is to train your own embeddings now with a wonderful Python library called PyKin that implements a lot of math behind link prediction methods. And we will finish with a demonstration of a use case for indication expansion for TLR7 inhibitor that we developed together with Idorsia. And we presented it in previous webinars, but here I give more focus on link prediction than we had opportunity in the previous webinar. Okay, let's start with introduction of biomedical KBHD. So biomedical KBHD is the largest knowledge graph of biomedical relationship data that is manually curated by expert scientists. And it captures relationships between lots of different biomedical entities like genes, diseases, chemicals, also drugs, variants, and contains over 24 million biomedical relationships in total. And it all is represented as interconnected graph of relationships that are all supported by scientific research. And those links are of very different types, so they are not just association links. They expand on the experimental context where the relationship occurred, but also on the type of relationship, like such as phosphorylation, ubiquitination, activation, and so on. So it is a very detailed information, and most valuable information, of course, is the causal effects that is also present, and it's between genes and diseases. So we have rich information about causal links between those two entities. So here is an example, a slice of data around EGFR. Like there are way many relationships of EGFR, but this is just to give it an example of how those relationships look like. So in GFR, like what is the neighborhood of it? So we have diseases like glioblastoma and small cell lung carcinoma. We also have some functions, apoptosis, proliferation of cells. And if you look at what context is available for each of the findings, for example, like let's look at EGFR and glioblastoma relationship. So we have a causal relationship, we have a direction, and we have an effect. So EGFR increases, you can say, phenotypical properties of glioblastoma. So it has positive causal effect on glioblastoma, and we know organism, and we know source of the information. So this is the content available in KBHD, and with this content already you can answer a lot of questions just through exploration, such as questions about genes. Um, what other genes your gene interacts with, what molecules are activated, inhibited, like what pathways the gene participates in, what diseases associated with certain genes. But you can also, of course, answer questions about diseases, associated molecules for each disease, biomarkers for diseases, and clinical trial information. However, there is more insights hidden in the knowledge graph beyond what can be extracted through just this superficial, you can say, exploratory analysis. And that's where we would want to extract it using more indirect ways, using computational methods. And one such method is a link prediction. So it is based on a model of understanding our knowledge graph as a network of biomedical relationships. But of course, this knowledge graph doesn't contain all relationships that are there out there in the nature. There are some links that are missing at random. Those relationships that haven't been yet discovered or haven't been published. And then the computational task can be finding those missing links, identifying those. And there is a so the task kind of can be mathematically formulated that given two entities in the graph, let's say like a specific gene, a specific disease, we want to predict whether there is this link exists or it does not. And one can so we can think about in terms of this task in terms of traditional link prediction approach using traditional machine learning methods. And so how would that go? You can design features to represent your gene. So you can describe your particular gene through its distance to through its neighborhood overlap with another disease. You can construct some features describing the relationships of this gene and disease. And then you can train a classical machine learning model like support vector machine or logistic regression to predict existing links in the graph in hope that it will also predict novel links. And it can work, but one obstacle with this approach is that it's not very easy to come up with meaningful features that would meaningfully describe very heterogeneous relationships between properties, between entities such as gene and disease. So then the question is, can we somehow automate it? And the answer is yes, we can. And it's done by utilizing link prediction methods that are based on node embeddings. So what are node embeddings? So let's say we have a knowledge graph here represented. We have node 1 and node 2, and we can learn a representation of each node in terms of multidimensional vector of numbers. So we develop a mapping of each node to some vector. So we try to encode all the information in this vector of numbers. How we do it, we'll talk about it later. And then what we are trying to do is that we can define in some sense a similarity between node 1 and 2 and node 1 and node 2 in any sense we want. We can say, well, node 1 and node 2 are only similar if there is a link between them. There can be other measure of similarity. It's our choice to define it. And then we want this similarity also to be reflected not only through links in our knowledge graph, but also within the embedding space. So we want just be able to take those two vectors and do mathematical operations on it and see, well, if we do this math and the value is such, well, and then we can call them similar, and it's similar in the same degree as they are similar in the knowledge graph. So we are trying to learn the representation of the structured of this knowledge graph in terms of vectors. And as you can see here, we are skipping the feature engineering completely because features are those numbers in the vectors, and they are learned through optimization procedure when those embeddings are learned. And advantage of this method is that those embeddings that we learn in this way are task independent. So once you learn them, you can use those vectors for link prediction. You can then use them for node classification, clustering, any task. And just want to give you intuition about what node embeddings are, what embeddings are, because yeah, it can be a bit overwhelming. What are those lists of numbers? So embeddings represent entities. In our case, nodes, and we can interpret those numbers in the vector as a position in a high dimensional space. So those are coordinates. So if you have an entity A and it can be then represented as a directional vector that points to some point in multidimensional space, and you have vector B, then you can call them similar if they are very close together. And you can define that similarity as a cosine of angle between those vectors. And it can be computed very efficiently through a dot product by multiplying coordinates of those two vectors. And so the similarity would be equal to 1 if vectors are similar, and will be 0 if they are random. So then they will be just orthogonal in some of the dimensions. It just will happen that in some of the dimensions they will be orthogonal, and the cosine similarity will be close to 0. And now that we know that, let's go get a bit in more detail in description of this task of training or learning node embeddings. So our task is to map those nodes in such a way that similarity, as we define ourselves, it's our choice, translates very well. Similarity in the graph translates very well to similarity in embedding space. So for this task, we need to define several things. We need to define similarity function in the graph. So taking the function can be presented like this: given two nodes, node 1 and node 2, what is the number that describes its similarity? How you compute it? So there should be an algorithm or a description of a way of computing similarity between two nodes in the graph. And I will give examples of those functions later. But you can, for now, let's say it can be just distance, for example. That could be one example of similarity function. How far is node 1 from node 2, minimal path. And it doesn't use node attributes, those similarity functions. So it only uses graph structural features, graph structure. Then we need to define what's called encoder, is a method or a function that maps a node in a graph into a vector. This is also on us as kind of data scientists to decide before we do before we try to apply node embeddings methods. And the simplest way is to use matrix lookup. So we can assume there is a matrix that we learn of node embeddings, and then we just pick a column that corresponds to a particular node. I'll cover it more detail in later slides. And then we want a decoder, a function that, given the embeddings, so vector representation of a node, can take those, can take two vectors representing nodes and give a similarity function estimate in a graph space. So, and the most common choice is the dot product, so the angle between vectors. And then task, like what are we doing during the learning process or the training process, is we are optimizing the encoder. So we are learning the matrix that represents, that captures all those embeddings. So that similarity in the graph, so this function, translates to similarity in the embedding space as closely as possible. And this is done by iterating through steps and adjusting values in the encoder matrix. Result of this is we get task-independent embeddings that we can use for link prediction, classification, different tasks. So now let's look at similarity function. So again, like we are very free to choose how we define similarity, and it can be dictated by, and should be dictated in some way by biology or what you're trying to do. But there are some very common methods, methods that are based on distance between two nodes, by neighborhood proximity, that are based on random walk. Node to vec is one common example of this. Then it's possible also to define similarity a bit indirectly as a similarity in terms of similarity in expression regulation pattern. And if you've seen one of the previous webinars called Leveraging the QIAGEN Knowledge Graph and Causal Embeddings for Insights into Drug Repurposing, that Andreas Kramer, my colleague, did several weeks ago, that's what's been used there. The similarity was defined in terms of expression regulation pattern. Or you can also simply define similarity in terms of existence of a link. So it would be very simple. It would be just a binary feature or a binary property. Well, if node 1 and node 2, if there is a link between them, then it is then similarity is 1. If there is no link between entities, then the similarity is 0. We can define it like that. Yeah. And then let's look at encoder matrix. So what is this matrix? So you've seen our feature vectors, node vectors, and now if we stack them next to each other, and this column dimension, we can say it represents just different nodes, like node 1, node 2 in the graph, node 3, like node n. Well, we can, and in rows we have dimensions, certain number of dimensions that we set. Then we get this matrix. And so the encoder itself could be just as simple as, well, your input is node 3. Well, how do I encode it? Well, I just look at column 3. That's my result. This is my encoding. Of course, the question is, where do we get this matrix from? We learn it in optimization process. And yeah, decoder is another choice we have, but most commonly it is a dot product, which corresponds to angle between vectors in high dimensional space. And it's also, it's very easy to compute. So this is a formula. So you just multiply corresponding coordinates and sum them up. Yeah, and the optimization. So yeah, we saw that we have this matrix lookup, but where does this matrix come from? Yeah, that comes from optimization process. We optimize an encoder, this matrix, so that similarity then in that we get through decoding um, to you can say through decoding, yeah, and computing, for example, dot product matches very closely to similarities that we defined in the graph itself, based on nodes, not on embeddings. And this optimization task of learning node embeddings can be formalized in terms of matrix factorization. And here's like an illustration how it can look like. So matrix factorization is decomposition of one single matrix into a multiplication of several matrices. And it allows for very efficient computations due to efficiency of modern hardware for computing matrix multiplications and generally being very efficient on with matrices. So here, let's say we have this graph ABC, and here it's actually not a matrix, it's a tensor. So we actually add one more dimension here that encodes different relationships. So it is a more more advanced model that is seen here, but we can also just look at the front slice of this cube. And how do we represent the graph? As I said previously, we can represent similarity as just as the existence of the link. So let's say, is there a link between A and B in R1? Yes, and we can see there is one here. Is there a link between B and C of type R1? No, there is not, and we can see there is 0. So this is a matrix that we would like to learn to reconstruct. And what we are trying to learn is a representation of B like entities or and the encoder, the vectors for B, C, A, all entities, and also like relationships in this case because it's a more advanced model. Where once you calculate, once you decode those, you get as close as possible to the original matrix or tensor, like or to the original similarity defined in the original graph. And it takes several iterations because like of how it works computationally. And of course, we never get like one-to-one. It's, you can see it will be like 0.9, 0.8 because it has to balance out a lot of things, because usually you take a limited number of dimensions in those vectors because you don't want also to overfit your model. So you typically go actually for 200, 80 to 200 the dimensions when you do node embeddings. So that's where kind of this lossiness is also coming from, and you kind of hope it generalizes and learns some patterns in the graph. There are of course some limitation in node embeddings in application of those methods, and I think it's important to mention those. Once you train on embeddings, you train only this matrix that encodes nodes for nodes that were exposed during training. So if you add new nodes to the knowledge graph that are not in the training set, there is no way of integrating them or having node embeddings for them without going through the training process from the start. Also, this method doesn't allow us to capture information from nodes, node attributes or edge attributes or like common graph features. So everything comes from kind of structural features of links and yeah. So how an entity structurally sits within the graph. One can, of course, encode properties more like in an RDF format where you externalize them and represent them as relationships. So that's an approach. But also another way like to deal with that is to use graph neural networks where it's possible to utilize in code node and edge features, but it's more computationally heavy compared to node embeddings. This is the node embeddings. This is something that doesn't require as much computation. It's more scalable. And yeah, so also there are still limitations also for scalability in terms of node embeddings, because if we saw this embedding matrix, it's just linear. It's number of dimensions, but by number of nodes. And and at some point it's just way too big matrix for common hardware to handle. So it's generally scalable to about 1 million nodes. So this is about the limit where node embeddings, then you have to have special hardware for that, for learning them. So now I hope to show you how to practically to train embeddings, and hope it gives an impression that it is easy and something that you will feel confidence in trying yourself. And now it's very easy with existence of a library called Pykin, which means Python knowledge embeddings, and it very popular package, over 1,000 stars on GitHub, very nice to use, very well supported, and it does a lot of math and computation for us. So here's a link if you want to check out. You don't have to remember it now. And the model, like the choice of parameters, encoder, decoder, and so on, that we're going to be using is called complex, and it's based on a paper called Complex Embeddings for Simple Link Prediction from 2016. There is some differences from like some additional things from that kind of link prediction on node embeddings training tasks that I described in the theoretical part. For one is in a complex model, embeddings, the embeddings vectors are now not real numbers, they're complex numbers. That's one change. And with this, why is it useful? And allows us to capture anti-symmetric relationships. And it's very important if you want, if directionality in your graph, if you have directionality in your graph, so you have causation in one direction, causation like phosphorylation in one direction or another direction, and you want to, for this information to be captured when you train embeddings, you cannot do that with real numbers, because then it's symmetrical decoder. So if you swap node 1 and node 2, the metric will be, the similarity metric will be the same. But having complex embeddings allows us to have anti-symmetric or asymmetric decoder. Yes, and so this paper is from 2016, is very theoretical. When this method became popular is I think it's 2020 with a publication, preclinical validation of therapeutic targets predicted by tensor factorization on heterogeneous graphs. In that paper, authors used this complex model to predict clinical trial successes and identify therapeutic relationships. And we will try the same today with the same method on our KB-HD knowledge graph. We have to start with creating train test dataset. So what we can include in it, or what we include in it, is molecule-molecule relationships, all of them. Complex and group memberships of molecules. We include disease and function ontology, and we include causal molecule disease and function relationships. And this is also what we're going to try to predict and evaluate on. So the way the kind of data set will look like, we just have entity one, its relationship, entity two. Another entity, its relationship with another entity ID. One has to be very careful when creating train test dataset on a knowledge graph. There is a lot of overlapping knowledge or knowledge at different levels in ontology, and you have to be very careful to dissect out things that can in the training set that might hint into things what happens in the test data set. An example of this could be causal relationships with Alzheimer. Well, if you have causal relationship with Alzheimer, then it's quite probable if you put it into a separate, or if you include, if you have a causal relationship in Alzheimer in a trained data set, and you have, let's say, causal relationship with familial Alzheimer in a test data set, well, it's quite trivial and not really interesting because they're kind of the same thing. So we want to exclude ontologically close relationships and maybe only capture one of those in a training test data set. So we do that to reduce data leakage as much as possible. Result is we have 1,600 test relationships and around 10 million training relationships. The number is so skewed is because we're going to be doing evaluation on a specific subset of causal molecule and disease relationships, because we want to replicate that paper, this paper from 2020. That was based on a very limited number of diseases. So that's why there is such a big skewedness between test dataset and trained dataset numbers. And this is how easy it is to train knowledge graph embeddings. Once you have prepared your train and test datasets, this is all the commands. You import PyKin, you specify train, test, TSVs, you specify model, complex embeddings that are antisymmetric, and so we can capture directionality information. You specify number epochs, so how many iterations of how many times the whole data set should be exposed during the training to the optimization process. So kind of good, like I've seen like 500 is a good number. It's very expensive. Here we do 10, but it should definitely be more than 2. And just to give an estimate what it takes to like computation, what kind of resources you might need. It's possible to do on CPU. It will be a bit slow. So I would recommend to use something with GPU acceleration. And it's quite easy nowadays with AWS and SageMaker, where you can just launch a notebook in AWS with this instance. I think it's about $1.50 per hour. It has NVIDIA 100 GPU. Yeah, so how many dimensions actually we are training here? So default value is 200 in this model. So each node will be represented as a vector of 200. 200 complex numbers, which translates to 400 real numbers. In terms of time, on this system, on SageMaker, it would take around 15 minutes per epoch. So you can, train, have node embeddings, train them within a day easily for 10 epochs.
Hey, Dimitri.
Yeah.
We have a question in chat that I would love to ask right now. Have other embedding approaches been benchmarked on QIAGEN's KB? Is complex the best performing approach?
No, it hasn't. But there has been a comparison of different embedding methods like complex, trans-E, and so on. I believe there is a AstraZeneca paper on that. Paper from AstraZeneca that did such benchmarking, but they did it on a different knowledge graph than ours. But complex, I believe in that paper didn't perform well. So like I'm a bit unsure like right now, like which method looks best. It looks a bit, you have to play around with it. So, but it will be interesting, of course, to try different methods and compare them. We chose complex because of the 2020 paper. It seemed like a good candidate.
Great, thank you so much.
Yeah, and once we finish training, we can save. Yeah, we just specify where we want to save the embeddings. I think they will be like around 1 GB in this case. So now, how do you do link prediction? What do you do with those embeddings? So our goal is actually to discover new knowledge or some hidden knowledge that is not we haven't seen. So we want to do link prediction. So we start with loading the model. We just point to where the model is, and then you can actually now do inference or prediction on CPU, because training is the most computationally intensive process. Decoding, so computing the dot product is not that computationally heavy, so you can do it on CPU. No special hardware required here. And so you have to load mappings of IDs used in embeddings to IDs in knowledge graphs, but yeah, it's a minor detail. Yeah, so now we can do the interesting part. We can predict targets. So how is it usually? This is a very common interface. So we call it head. We have a relationship, and then we have a tail. So like this is, we can call it a head node, a tail node, and a relationship. And how link prediction usually works is that you fix a head. So you specify, okay, for this entity specified here, and this relationship, causal relationship, what are my candidates that, so predict me, compute me all similarity scores for this triples for where this can be like, where a tail entity can be anything like wildcard. It's possible, of course, to do a combination, a completely free-fall link prediction where you just any starting entity, any relationship, any tail entity, It's just too computationally heavy. Usually, you know, like what do you want to predict a kind of a partner to. So in case I don't remember what this necessarily encodes, but let's say it's NO1 gene. Like, what are causal relationships with what diseases are causally related with NO1? And it just allows us to constrain the prediction, so you get results much quicker. Once you compute this, you get the output like this. So what do we see here is a list of tail entities. And well, here is actually ingenuity IDs for tail entities. And here we see scores, complex scores, in how likely there is a link of our fixed entity with this particular tail entity, and it's sorted in a descending order. So this would be the most likely. So this would be the output, and then you can work with that. Then how do you usually evaluate your embeddings? Well, because we saw this was a train and test data set. The common metric used for evaluating link prediction is like embeddings for link prediction tasks is what called hits at n, and can be like n where n is number. It can be, for example, 10. And it is a number that shows how many true relationships do you recover in top 10 rank results. It can be like 1. It's quite low usually. So it can be like in hundreds of so it's kind of fraction of 200s or 300s, so that will be regular number. And then you can also not necessarily represent it as a number, you can also plot a graph. So let's say we again we have this sorted list of predicted tails relationships, or predicted relationships, where here is the most likely, and this is like less likely relationships. And then we can see how the trend goes of how many of true relationships have we observed now that we like fetched 100. Like if you take top 100 predicted relationships, how many true relationships have we recovered? 75% in this case. So it's kind of accumulative curve. So then we can plot this curve split by different disease categories, and we can also compare it to a node degree based predictions. So those are very so node degree based predictions is a way kind of controlling for biases in our graph, because knowledge graphs, they don't represent they are biased by scientists, by the research they're doing, because there are certain areas that are scientists like, is it over research, you can say. There are more relationships related to certain things, and then it's just the way embeddings can be, they can, and that information, this bias, you can say, can seep into node embeddings, and it's not really biologically relevant. It's not really biologically interesting that there is more research about EGFR. The link between EGFR in glioblastoma is more likely just because it is a very popular topic or a very well-funded topic. There is no biological relevance to it, and we want to see how much kind of biological relevance is captured by node embeddings compared to just biased kind of node degree based approach where we predict just based on how many existing relationships an entity has. So here the solid line represents our link prediction curve, and dashed line, this is no degree based prediction. So kind of this area between dashed line and solid line would be indicate how much of biological insight the node embeddings capture in them, and how much you should maybe trust link prediction, how much you can rely to link prediction in certain disease areas. So this can be a way of again evaluating your node embeddings. I think it's very important to know to eliminate this bias. So now I want to demonstrate you a more practical, a real use case that was previously presented at a webinar that we did around half a year ago called Supercharge Your AI in Drug Discovery with high quality biomedical data that we did together with Idorsia and Neo4j. So I'll just, I encourage you to watch that webinar if you want to know more details. So because I will not be covering all the details of our findings in it. But in that webinar, we walked through a use case that explores TLR7 as a potential drug target. And TLR7 is a receptor that sits within cell and detects single-stranded RNA, and single-stranded RNA kind of an indication of virus infections, and then TLR7 once it detects it, it initiates immune response. And a lot of research in previous 20 years has been on how TLR7 does it. And there's been also findings that severe COVID is associated with loss of function in TLR7. So we can say like TLR7 in its homeostasis in a normal condition, it ensures antiviral immunity. Loss of function, you get, it undermines your ability to detect infection, and you get severe COVID. Gain of function, well, then you get overreactive chronic inflammation, activation of immune system. And also like in recent 2 years, there's been established a causal direct link between TLR7 and lupus, autoimmune condition. And this is very typical of kind of pharma use case where you read a paper and find new associations. You see if you can develop a drug for it, and want to see how from the data you have before you jump into like real development, you want to gauge how much it makes sense from the data perspective. And we can start this by just doing some exploration in knowledge graph. We can we can take TLR7 as our gene. We can see how many causally related diseases are there to TLR7. We can see also what type of drugs exist in clinical trials, and how many activated drugs and how many inhibition drugs for TLR7 exist, and for how many disease indications. So this is like a kind of beginning exploratory approach, right, for all of this. And then you can also dig deeper into this. You can split your clinical trials into approved and in development, and again, like activation and binder. And so what you can see here is that in activation, we have a lot of, the intention is to boost immune system for these diseases. And in inhibition, we see in development, we can see it's very thin. There's not so many, and it's yeah, SLE as kind of indicated by the maybe actually initiated by the paper that was published. So autoimmune diseases. So, and of course, well, as if you're a pharma company, you don't want to go to places where others are already looking into. You want to maybe do something new. You want to explore new indications. So where should we go then? And we can use BKB and yeah, our link prediction approach, computation approach to see if we can come up with novel indications. We can predict them and see if they're like less obvious things but that also have good like supportive evidence for them. So yeah, let's just do that. Let's just take TLR7 and do link prediction that we just and for diseases and for which yeah causally related diseases are there with that TLR7 promotes that we can have an inhibitor we can utilize an inhibitor for. And we get this list. And it's just like will be like if you imagine if it was not color coded, it would be like a huge list. Like how do you make sense of it? You can map those disease and functions in ontology, and then you can start seeing some patterns here. Something that supports maybe like encephalitis, immune response to brain, something that's like maybe indicative of neurodegenerative disorders or neuroinflammation. That's how you can make sense of your link prediction results. Then, additionally, you can do something more advanced. You can also include not only so you have to also of course filter known predicted results. You can also include those known results and see and cluster your results using UMAP based on proximity in a protein-protein interaction network. The details of how it's done, again, I would refer you to the webinar. But let's say we do that. And now we can see this cluster that really stands out from all other, and it's a novel cluster. So it's new things that we predicted. And again, we can see some patterns here indicative of nervous system inflammation, CNS. So again, this is kind of some kind of new biology that we see because it's really separated in protein-protein interaction space. So this is very interesting here. And additionally, you can also try to understand biology behind this cluster. Again, I'm just offering you like an alternative approach, computational approach to look at those results. Is to use word clouds on a list of enriched pathways and functions. So we take those diseases, we do enrichment analysis for connected pathways and functions in each cluster and do word cloud. And we can, again, kind of already like immediately pick out some patterns here. So we can see like lung, we have disease, inflammation, liver. Yeah, we have also inflammation here, but we have encephalitis. Yeah, a lot of encephalitis. So there are common things here in those novel and known classes, such as inflammation. But we also have differences here, and one such difference, yeah, I would say kind of relation to like a new organ, to a brain. So again, indirectly kind of indicating towards the fact that it would be interesting to look like in some neuroinflammatory condition as a potential target for TLR7 inhibitor. Additionally, we've been predicting links in as if we were thinking about we were developing inhibitor, right? So we were trying kind of to suppress a disease. It was a causal activation of a disease, of TLR7 that we were trying inhibit. But we can predict links in the opposite direction. We can predict causal decreases. So like why? Well, one meaningful thing you can do with that is you can potentially think about kind of potential side effects or disruption in signaling pathway that might occur after application of TLR7 inhibitor. So yeah, if we do that, and here like there's a bit of skipped steps, and we do a bit of enrichment analysis, then we can see, and quite predictably so, that, well, if we do inhibit TLR7, we disrupt our protection against viral infections, cancer, bacterial infections. So that's kind of also what we've seen with severe COVID. Inhibiting TLR7 can be have those implications. And yeah, so with that, we're kind of summarizing this use case, kind of once you kind of did those all those analysis for TLR7, yeah, you would, based on those results, you would then consider some kind of neuroinflammation, neurodegenerative disorders like Alzheimer as an interesting indications to explore further. Yeah, as with this, we're about to finish. I hope so today what we did, we learned fundamentals of node embeddings for knowledge graph. It's a way broader topic to cover in 1 hour, but I hope it was a very understanding introduction, understandable introduction, and to kind of maybe will serve as a base for you to learn more. We showed how to apply PyKin to train your embeddings. And just here, I hope kind of to break maybe a barrier for you that you feel more empowered to try it on your own to apply those methods with how, again, with the way I showed you, it doesn't really take a lot of code to do that. A lot of course, yeah, there are a lot of things we would need to do in creating train test data split, but the mathematical computational part is abstracted very well right now by this package. And we looked at a practical use case of indication expansion for TLR7. Here is a list of resources that I would like to recommend to you. Is a webinar, two previous webinars we did. One is, the first one is covers this TLR7 inhibitor, TLR7 use case. The second one was done by Andreas Kramer, and there were different types of embeddings there used for different purpose. But again, I highly recommend if you're interested in this subject. Then PyKin package itself. Then, if you're interested in machine learning with graphs and kind of theoretical background behind traditional link prediction methods, node embeddings based ones, and methods that use graph neural networks, I highly recommend this Stanford University course. Kind of, I was watching it myself before this presentation. And these two presentations for complex embeddings and where it was first utilized to do preclinical validation of therapeutic targets and predicting clinical trial success. I think so those are two references I would like also to highlight. With this, I would like to thank you for attending the webinar today. And if we have time for the questions, I can answer them now, but otherwise we'll try to answer them after the webinar in the email.
Thank you so much, Dimitri, for such a wonderful presentation. We did get some questions. We'll take a few of them here. And just like Dimitri said, please reach out to us if you have any further questions or would like to learn a little bit more. All right, so first question would be, how do you handle noisy or incomplete data when performing link prediction on knowledge graphs?
Yeah, I would say that you want a high quality data when especially when working with machine learning approaches. There's a common saying, garbage in, garbage out. So the best way is to use clean data. So human curated high quality data would be an advantageous if you want high quality results, because it's already a computation, as you saw. Like even with high quality data, you get not all the results are guaranteed that there is a link. It's all about probabilities. So you just, with noisy data, you're just increasing the probability that there will be more irrelevant results in your link prediction.
Gotcha. Speaking of data, how does BKB-HD ensure data provenance and transparency, especially when it's used in critical applications like drug discovery?
So all our BKB data, biomedical knowledge-based data, has associated source finding information. So all the relationships have as an attributes information about the source, publication, database. So this all can be traced, and yeah, the curation team takes care of that. That this is consistent, correct, updated, up to date, and so on.
Excellent. One more question, since we are running a little bit over. How easy is it to integrate external data sources for enriched analysis?
So I assume the question is about integrating with biomedical KB HD.
Exactly. Yep.
Yeah. So then, yeah, it should be quite easy because all our entities, such as genes, diseases, they have mappings to external database identifiers. So yeah, it's of course there is a depends on what data you're integrating with. Fitting it into ontology might take some effort, but otherwise, like, I mean, the kind of key anchor points are all there. So there is all the nodes have an external database identifier, so it's kind of, you can hook up onto them and merge the data set. So that should be quite easy. But there is always like devil in the detail.
Excellent. Well, once again, thank you so much, and we hope to see you in future webinars. Take care, everybody.
QIAGEN.
Sample to insight.